So for general physiology na lecture, we started with membrane physiology. We discussed there about the membrane transport, diffusion, passive transport, active transport, and on what is the significance of the permeability ng cell membrane when it comes to the maintenance of the percentages, diba? That would make your cells live. Okay? Kumbaga, it will maintain the health of the cells. And then next to that, we studied about the membrane potentials and the action potentials, specifically for nerve and muscle cell membranes. So as I've said sa mga previous lecture, we are trying to make sense of each physiologic mechanism as we go along from one topic to another. And for today, this will be our last topic for prelims. Ang next topic natin is about synapses and synaptic transmission. So first, before we before we start the discussion, watch this video first. Okay, so we have the nervous system. It consists of neurons, okay, numerous neurons. It carries with them signals, no, or impulses from the membrane potentials. These are to be communicated to one another to perform its desired function. So last meeting, nag-discuss tayo how these signals are propagated, di ba? Action potential from the dendrites, bodies, down to your axons. And you have the influx of your positive ions, the sodium ions, to depolarize the nerve membrane and it will elicit the action potential, di ba? So as the action potential propagates, it will then reach this part of the axon. We call this axon terminal. Okay, this is the terminal of the axon. So what then happens if the action potential reaches that point? So that will be discussed now. And we will discuss what synapse is and what happens in synaptic transmission. Okay, so what is a synapse? A synapse is a specialized junction where impulses are transmitted from one nerve cell to another cell. So when we say another cell, it can be another neuron. So for example, from axon terminal to dendrite, we call that axodendritic synapse. If it's from an axon to cell body, we call it axosomatic. When we say soma, that is another term for your cell body. And if it's from an axon to another axon or along the length of an axon, it's called axo-axonal synapses. So that is another cell, no, from one nerve cell to another nerve cell. Yun yung tatlong pwedeng synapses: axodendritic, axosomatic, or axo-axonal. Another cell, okay? Another cell na pwedeng magkaroon ng synapse is between a nerve cell and a muscle cell. So from an axon terminal to a muscle, we call that neuromuscular junction. From a nerve to a muscle, neuromuscular junction. And we'll discuss also, we will also discuss that at the latter part of this lecture. It may also be a nerve to a gland cell. So for example, you have your salivary glands, you have your sebaceous glands, you have your sweat glands. So it can also be from a nerve to those glands. What else? Sensory receptors. So for example, from a nerve to the skin, we have the cutaneous nerves. So those are the different areas or locations where synapses can be found. So nerve to another nerve, nerve to a muscle, nerve to a gland, nerve to a sensory receptor. And then for nerve to nerve, you have the following examples: axodendritic, axosomatic, axoaxonal. It will depend where the synapse, kumbaga, what are the two cells where the synapse is in between. So wherever in the body that a nerve cell comes in contact with, there ought to be a synapse. So basically, this is how Basically, this is how communication happens within the nervous system. So as you've seen kanina sa video, no, parang from electric impulse from one nerve cell to another. So those are action potentials, and we jump from one cell to another in terms or in by synapses. Yan. Kumbaga yun yung pedestrian lane from one cell to another. So you're already familiar with the three parts of the neuron, no? So ang dendrites, it conducts impulses towards the cell body. Ang cell body, it's the nucleus containing part of the neuron. Ang axon, it conducts impulses away from the cell body. So there are three components of a synapse: the terminals of the presynaptic axon, a target on the postsynaptic cell, and a zone of apposition. So meaning of zone of apposition, this is the proximity of the two structures. Basta as long as may presynaptic and postsynaptic, may pinanggalingan, may pinuntahan from one to another. So at the middle, that's your zone of apposition. So there are two types of synapses based on the zone of apposition, either electrical or chemical. So first, we have the electrical synapse. Okay, so for the electrical synapse, these are the characteristics. So there is a close apposition of pre and post synaptic cells at gap junctions. Okay, so you call this gap junction in between the two cells, the presynaptic and the postsynaptic cell. The current from presynaptic cell directly enters the postsynaptic cell. Direct. And there is continuity of cytoplasm from pre to post. So ganito sila ka-close. Okay? That's for electrical synapse. But for a chemical synapse, there is what we call as a cleft. Okay? Kumbaga, there is a bigger apposition. The pre and the postsynaptic cell are separated from one another with a cleft at the middle. Pag electrical synapse, they call it gap junction, and they are closely close to one another. Closely adapted ba? Or basta yun nga, there is continuity of cytoplasm. But for this, ang cytoplasm ng presynaptic is not continuous sa cytoplasm ng postsynaptic. Okay, there is that space. So there is a cleft separating the two cells. There is no communication through channels compared to this. And then the current leads to the release of a chemical transmitter. So we will discuss what happens in the synaptic transmission sa chemical synapse. Dito kasi sa electrical synapse, it's simply electricity and then passed to another cell. Pero dito, the electricity will just be a trigger for a lot of events to happen before it reaches the postsynaptic cell. So this is a tabular form of how we will differentiate the two. So as for the prevalence, majority of the synapses that can be found in the body is a chemical synapse. May electrical, pero kunti lang. So dito kasi kailangan rapid. When the synapse needs to be more rapid than the chemical, dun siya nag-electrical. Pero it's harder to regulate kasi pass lang siya ng pass compared to chemical that there is a processing. Distance. Okay, distance between pre and postsynaptic cell membranes. Sa electrical, 4 nanometer. Sa chemical, 20 to 40 nanometers. I will not ask you to memorize yung exact number. I just want you to take note na there is bigger distance kay chemical compared kay electrical. And this is, kumbaga, just imagine how much different they are. Next, cytoplasmic continuity sa electrical, yes, meron. But for chemical, hindi. Okay, there is a cleft at the middle. Types of induced response, pag electrical, automatic na excitatory. So meaning, action. Pag chemical, it can either be excitatory, action, or inhibitory, no action. So that's a major difference also between the two. Ultrastructural components. Yan. So sa gitna, it has gap junctions channels, gap junction channels, as you've seen sa figure earlier, the slide before this. And then for chemical, you have the following. So may presynaptic vesicles, may active zones, may postsynaptic receptors. So later, we'll identify that better. Agent of transmission sa electrical, ion current. Ion current. So from the electrical impulse kay pre, ion current, electrical impulse pa rin siya kay post. Pero for chemical, as I've said, no, electricity will just trigger it will just trigger a series of events, but it will use chemical transmitters for the synapse to happen. Speed of transmission, of course, yan. Since wala siyang cleft, it's faster. Ito, slower. But then, it's still fast. 2 milliseconds. Just imagine, no? 1 second, just 1 second, and then you divide it into 1,000, diba? Milliseconds. That's 2 lang. 2 milliseconds. So, it's very fast. Just for example, no, I'll just give you a quick overview. So I am thinking of what to say, and then the nerve impulses already reached my mouth to pronounce the words that I'm thinking. So just imagine, no, the transfer from one nerve cell to another, from the thought towards the action of me speaking the words. Kumbaga, nasa utak ko, kumbaga, ano bang English or Tagalog noon? What is the comprehension? Kumbaga, how will I explain it better in the understanding of my audience? Kumbaga, all of the nerve impulses are happening in the brain. And then you are putting it into action when you speak the words out. So just imagine the nerve impulses from thought to action. So saya. So kumbaga yan, there are a lot of chemical synapses happening at this very moment. Even while you are listening, no, the signal that comes to your ear as you hear me, and then you are processing in your mind the words that I am speaking. It's processing in your brain how you understand it, how you hear it. So those are all happening with chemical synapses. Masaya. And then direction of transmission. So pag electrical, mostly bidirectional. So kanina sa picture din, from pre to post, post to pre, they're free. But for chemical, lagi yan from pre to post, no turning back. Hindi siya babalik. It's unidirectional. So kumbaga, yan nga, in terms of higher cognitive skills or physiologic mechanisms, more on chemical synapses talaga yung majority that is happening. So there are parts of a chemical synapse. So you have the terminal button of the axon or the axon terminal. It's part of your presynaptic cell. So yan siya yung yan, that's the... So ito, what is this example? So you have here your dendrites. You have here your cell body or soma. Then you have here your long single axon na may axon terminals or buttons, terminal buttons. So from here, from axon to your dendrite. So what is this synapse? What is this? What does this exemplify? So this is an example of an axodendritic synapse from axon to dendrite of another nerve cell. So zoom in, these are the parts. Yan, axon terminal, then you have the here neurotransmitters in vesicles. So itong mga vesicles na to, these are found at the axon terminals. Kumbaga, they lie in wait for an electric impulse to come before they do their action. So kumbaga ito waiting area of vesicles with neurotransmitters inside the vesicles. Then you have here the synaptic cleft. Yan, mga 20 to 40, ano yung kanina, nanometer, parang ganyan. Basta yun, yung distance between pre and post. And then you have here the postsynaptic cell. So kapag check, itong mga parts na to, it's a chemical synapse. Okay. So now let's go to the first, ano ba? What happens in a synaptic transmission? Okay, first, presynaptic exocytosis of neurotransmitters. So presynaptic, it's the axon terminal. Exocytosis, when we say exo, it's, ano ba? Exo. Kasi pag endo, papasok. Pag exo, palabas. So goes outside. Exocytosis of what? Of neurotransmitters. So ito yung step by step. We'll run into this. So step number 1. Action potentials arrive at axon terminal. So we have studied about yung tawag ito? Sodium and potassium voltage gated channels, di ba? Na nasa along your axon, specifically nasa mga nodes of Ranvier. So from node to node, saltatory conduction, yung action potential runs along the axon until the action potential reaches your axon terminal. Okay, so imagine, nasa axon terminal na tayo. Step number 2, because of that action potential, it will open the voltage-gated, ano yung Ca? What ion is Ca? Periodic table of elements. Ca is calcium. Okay? Voltage-gated calcium channels will open. So when we say voltage-gated, the stimulus for its opening is electricity. So ito nga yun, yung number 1, yung action potential that reaches your axon terminal, that itself is the electrical stimulus which will open the voltage-gated calcium channels. Okay? So when it opens, so specific siya for calcium, so ang dadaan sa channel na yun are calcium ions. So number 3, calcium enters the cell. So, nasundan nyo pa. So, ito yung number 1, action potential. Once it reaches the voltage-gated calcium channel, it will open. And then number 3, from the extracellular fluid, yung calcium will go inside the cell through the cell membrane ng axon terminal. So, pasok si calcium. Okay, that's step number 3. Calcium enters the cell. Once the calcium enters the axon terminal, step number 4, calcium signals to vesicles. So that would be the knocker. Okay, knock knock, vesicle, I'm here. So the calcium will signal the vesicle to move towards the membrane of the axon terminal. So kumbaga, it will signal nga, kumbaga parang punta ka na doon, go there, vesicle, I'm the calcium. You listen to me, vesicle, kumbaga I'm here and I will tell you, command you to go to the membrane. So that's the action of your calcium, signal the vesicle to move towards the membrane. So number 6, dock vesicles release the neurotransmitter by exocytosis. So there, that's your docking protein. So kumbaga parang dun siya nag-dock. Di ba alam mo yung dock sa pier? sa mga ship, cruise ship, ganyan, or when you go to the port. So they're yan. Kumbaga nag-dock, mga yacht, wow, social. So when the ship or when the yacht go towards the port, wherever it goes, that's the dock. Okay? So, hirap. So, yon, the vesicles will dock doon sa membrane, and then it will perform exocytosis wherein yung laman ng neurotransmitters will be released from the presynaptic cell. So number 7, the neurotransmitter diffuses across the synaptic cleft and binds to the receptors. So this figure is what you call as the presynaptic exocytosis of neurotransmitters. Okay? Okay. So just a quick run through. Action potential arrives at axon terminal. It will open the voltage-gated calcium channels. It will cause the influx of calcium, which will signal the vesicles to dock at the membrane to perform exocytosis of the neurotransmitters through the synaptic cleft, and then it will bind to the receptors of the postsynaptic cell. Okay. This one. Okay, this figure tells us the synaptic vesicle cycle. What happens to the vesicle once it already exocytoses its neurotransmitters? What happens? It is being recycled. Okay? Kasi neurotransmitters lang nag-exocytose. The vesicles are still part of your presynaptic cell. So here, sige, yan natin. Run through. Okay? So you have here neurotransmitter up. Sige, reserve pool. Dito tayo mag-start. So reserve pool, these are lying in wait. Okay? As long as there are no calcium ions, it will stay as reserve pool. But once there is a signal from calcium ions, it will dock. Number 3, docking sa membrane. And it will produce ATP. This one is energy. Energy, yan, ATP. It will undergo priming. Yan. Ano ba yung priming? Number 4, ATP-dependent priming reaction. Basta it's part of exocytosis. And then, number 5, ayan, that makes them competent for calcium-triggered fusion sa membrane. And then yan, magfo-fuse yung membrane ng synaptic vesicle sa membrane. It will cause fusion, and then it will release the neurotransmitters. And then it is being recycled. Yan. So it will again receive neurotransmitters. Yan, neurotransmitter uptake, and will be part of the reserve pool again. Basta ganon siya. May neurotransmitter siya. Pag may calcium, it will dock and exocytose. Tapos, ito, endocytosis, meaning it will be formed back inside the cell. Hindi siya ang nag-exocytose lang are neurotransmitters, but the synaptic vesicles are endocytosed back to the reserve pool. It will receive again neurotransmitters and wait ulit sila doon. Yun yung kanilang trabaho. That's their work. So mechanisms for recycling, there are 3 ways how synaptic vesicles are being recycled. So first, reversible fusion pore. Sa reversible fusion pore, ano siya, yan natawag na parang kiss and tell. Wow, kiss and tell. So dock, release, tapos ano agad, mag-fuse agad, then back to being a synaptic vesicle. So ganun lang siya. Dock, release, then go back as a synaptic vesicle. And then yung clathrin mediated naman, ito, as you can see there, yung clathrin, it's basta parang siyang protein. Kumbaga, it helps in the endocytosis of your synaptic vesicles. Kumbaga, it has help. Yung reversible fusion kasi, wala siyang ibang help. No clathrin, it's not clathrin mediated. Pero kapag may performance or may help from clathrin na protein, then yun yung clathrin mediated. Kumbaga, nag-fuse totally yung membrane of your synaptic vesicle sa membrane ng axon terminal. So that's why kapag nagkaroon ng clathrin dito, that's when mag-endocytose ulit, magkakaroon ng sariling membrane, si synaptic vesicle to be recycled. Kapag bulk retrieval naman, ayan, kumbaga hindi na na-maintain yung shape niya na circular. Okay? Parang ganon. Pero basta ano siya, bulk retrieval. Basta, ganun. Tatlong ways. Okay? 3 ways for recycling, no? Reversible fusion for clathrin-mediated and bulk retrieval. In any of the ways, ang general principle is that synaptic vesicles are being recycled to receive neurotransmitters again to become part ng reserve pool. Yun. Perapan ko yung sarili ko, na. Number! Yeah, next pala. Not may number. Next. Sabihin ko sana yung slide number. Anyway. So, what else? Ito. This is also a noteworthy part. Okay? So, ito, zoom in pa siya na in between what happens between your synaptic vesicle and your membrane ng axon terminal. Part pa rin ito ng exocytosis. So this one, and ito, sabi ko nga zoom in, okay? Sige, isa-isahin natin yung parts, no? So we have here, yan, vesicle membrane, then here is your plasma membrane, this is your extracellular side, this is your cytoplasmic side. So sige, isa-isahin natin. First, we have the neurotransmitter transporter. So itong neurotransmitter transporter, ang ano lang yan, ang function is for the neurotransmitter uptake. Kumbaga, that's the door for the neurotransmitters to go inside the vesicle during recycling. Yan. And then this one, yan, ito, take note of this part. This is what you call as your synaptobrevin. Itong synaptobrevin, hindi. Wait lang. Itong synaptobrevin, yun yung color red. Okay, tiga ganito Okay, yung color red, mag-color muna tayo. Itong synaptobrevin or VAMP, that's another term, okay? It's the color red dito. Itong SNAP25, ito yung color blue. Tapos yung syntaxin, yun yung green. Itong SNAP25 and syntaxin, those are part of your plasma membrane. Ang synaptobrevin, that is a part of your vesicle membrane. So kapag ikot-ikot si vesicle and it reaches si membrane, it's like a jigsaw puzzle that they will find each other. Now once si synaptobrevin ni vesicle, na-meet niya si SNAP25 and si syntaxin of your plasma membrane, they will form the snare complex. You see here, snare complex, S-N-A-R-E complex. And then this will pave the way for vesicle fusion to happen. Okay? Kumbaga dyan sila nag-meet and then they shake hands and then go here, dock here, we're good friends, ganon. So kumbaga that's their way of meeting up. Okay? Yan. It results from the tight interaction between synaptobrevin in the vesicle membrane and syntaxin in SNAP25 in the plasma membrane. Tapos ito, yung MUNC18, it interacts with the snare complex essential for vesicle fusion. So kumbaga may external factor pa na, uy, sali ako. Pa-join, ganon, si MUNC18, si MUNC18. Okay, so yun na. Again, neurotransmitter, it's for neurotransmitter uptake. Part siya ng vesicle membrane. And then si synaptobrevin kay vesicle membrane siya, si SNAP25 syntaxin kay plasma membrane siya. When they meet, they became good friends and they called their group snare complex. Si MONC18, nakisali for vesicle fusion. Okay? Next, yan. Ito naman, synaptotagmin. It's part of your, these are proteins mga proteins. So, yung synaptotagmin, it's part of your vesicle membrane and this one serves as a calcium sensor. Okay? Calcium sensor for exocytosis and may also interact with, anyway, okay na yon, calcium sensor. Kasi di ba kanina, the calcium signals the vesicle membrane to go to membrane. So yan siya, si synaptotagmin ang nagde-detect, nagsisend yung sensor kay calcium na, andyan si calcium, let's go to the membrane. Ganon. And aside from that, may calcium channel din dito and it also, kanina, di ba? Calcium-mediated fusion. So, calcium is also needed for the fusion interaction. So, basta taga-sense kay calcium, synaptotagmin. What else? Synapsin. Ayan. Ito si synapsin. It is important in regulating the availability of vesicles from the, important in regulating the availability of vesicles from the reserve pool. So, para siyang name tag na, part ako ng reserve pool, you can tap me anytime. I have my own neurotransmitters inside me. So, ganun si synapsin. It's important for the regulation ng availability. So, ito, okay na yun? Yan. So, yan. Ito, sorry. Ito, neurotransmitter uptake. Ito, vesicle fusion. Ito, calcium sensor. And then this one, availability sa reserve pool. So, take note of those proteins. Ito, hindi ko siya ma-explain, pero part siya rin ng fusion. Pero sige, wag niya nang anuhin. Hindi ko alam ibig sabihin ng RAM eh. Or if you want to read more about it, it's up to you. Pero sa discussion ko, goods na sa akin itong synapsin, synaptotagmin, snare complex, tsaka neurotransmitter transporter. OX na for me. Next principle, we have synaptic delay. So kanina kasi, yun yung difference, di ba, of your electrical synapse and chemical synapse. Na ang chemical synapse ay may synaptic cleft and it takes longer because of the distance. Pero super minimal nga lang. Sabi nga dito, ang synaptic delay is just 0.5 milliseconds. Just fast, so fast. So it's the minimum time required for transmission across the synapse. So what happens dun sa delay na yon? Itong mga ito. Ay, sorry. Cited. Okay, so what happens in a synaptic delay? There is discharge of transmitter by the presynaptic terminal, diffusion of the transmitter to postsynaptic membrane, action of the transmitter on its receptor, action of transmitter to increase membrane permeability, increased diffusion of sodium ions to elicit postsynaptic potential. So yun, kaya siya matagal dahil sa those are the events that happens in that 0.5 millisecond na synaptic delay. So kapag sa electrical, this does not happen. Or kung nag-happen man, it's very much, mas faster siya. Pero hindi wala siyang transmitter pag electrical. Anyway, for chemical synapse. Okay. So yun, medyo na-visualize niyo ba what happens? So technically, we're done with the part na presynaptic cell. So basically, yun na yung nangyayari from action potential hanggang sa nagkaroon ng exocytosis and then nagka-recycling ng synaptic vesicle. So next part, yung synaptic delay, syempre yun nga, yun yung nangyayari. So diffusion, tapos action of the transmitter sa receptors, yan. So ngayon, next topic are the types ng receptors that you can find in a post-synaptic cell. It can either be ionotropic or metabotropic. Yan yung two types of receptors. So kapag ionotropic, it's a type of a ligand-gated ion channel. When we say ligand, chemical. 'Di ba kanina na-mention natin yung mga voltage gated eh. Yun kasi yung two types of channels. Yung mga channel proteins, it's either voltage gated or ligand gated. Kapag voltage gated, ang stimulus niya is electricity. Kapag ligand gated, ang stimulus niya is chemical. So in this case, yung ligand gated ion channels, these are stimulated by the attachment of a chemical. Specifically, you call it neurotransmitters kasi galing siya from a neuron. Galing siya sa synaptic vesicle from an axon. So yan siya, axon, tuloy ions na nagdi-diffuse until they find their spot sa receptor. So the neurotransmitter binds and it will cause the ligand-gated channel to open and then the ions will flow across the membrane. So yun yung ligand-gated. Now let's go to the other type of receptor called the metabotropic or the G-protein coupled receptors. Ito naman, ang idea lang niya is, it utilizes another protein aside from the ion channel itself. So dito, may natawag na G-protein. Yun lang siya. Kumbaga may mediator, may other cast siya. May other part ng team. So dito sa G-protein coupled receptors, the neurotransmitter binds to the receptor ng G-protein and the G-protein is activated, yan yung mga alpha, beta, gamma units, okay? And then they will send intracellular messengers, yan, may mga effector protein, and that will signal or give yun, modulation sa ion channels to open. Okay? Kumbaga may iba silang manager. Ito kasi, they manage themselves. Kapag may nag-bind na neurotransmitter, I will open. I'm a ligand-gated ion channel. Pero this one, sa G-protein coupled receptor, meron kang manager. Kumbaga sa artista, you have, kumbaga may dadaanan ka muna before I do the project. Parang ganon. Okay? So G-protein subunits or intracellular messengers modulate the ion channels. That's when the ion channel will open and cause the ions to flow through it. So basically, that's the difference between ionotropic and metabotropic. Wait lang, check ko baka may nakalimutan akong i-mention. Okay, nasabi ko na rin. Both types of receptors can either excite or inhibit. The sign of the signal depends on the identity of the transmitter. Okay, sige. So yun, either excitation or inhibition. So what do we mean by that? Yan. So ito, yan siya. Ito yung mga post-synaptic potentials natin. As mentioned earlier, it can either be an excitatory potential or inhibitory potential. So yan yung EPSP tsaka IPSP. Okay? Mga EPSP, those are excitatory post-synaptic potentials. Ang IPSP, inhibitory post-synaptic potentials. And na-mention natin from last week yung tinatawag na threshold potential. Na once na-reach ang threshold potential, that's when an action potential will be elicited. So, kumbaga ang nangyayari, this is your resting. Again, what is the resting membrane potential of most of the cells? Negative 90 millivolts. Okay? Negative 90 millivolts. So, kapag may dumating na synapse kay postsynaptic cell, pag excitatory, tapos may inhibitory din, tapos nag-excitatory, inhibitory, excitatory, excitatory, until it reaches the threshold, then that's the time na magkakaroon ng action potential and it will propagate to whatever postsynaptic cell that is. As mentioned kanina sa first part, yung definition of synapse, yung mga postsynaptic cell natin can either be a nerve, another nerve, it can also be a muscle, it can also be a gland, it can also be a sensory receptor. So kumbaga ano siya, it's a collective of all the potentials. Excitatory, inhibit, excitatory, excitatory, inhibit, excitatory until it reaches the threshold, tsaka magkaka-action potential. So it will depend on ano ang dumating. Okay? So excitatory postsynaptic potentials, it can result due to fast or slow channels. The goal is to bring the membrane potentials closer to the threshold. So ito, for example, ito negative 55 dito. Kasi, di ba na-mention ko rin from last meeting, no, from last week, na for nervous system, for nerve cell membranes, ang resting membrane potential nila is negative 70. Tapos ang threshold potential is 15 millivolts more negative. Kaya siya negative 55 dito. That's for nerve to nerve. Pero kapag sa muscles na, sa glands, ganyan, negative 90 na yun, karamihan. Okay. Yun lang, I just want to explain bakit negative 55 because we're talking about nerve cells here. Okay, so pag EPSP, what it does, it brings the membrane potential. Ang gusto niya mag-threshold. Okay? It will increase the likelihood of generating an action potential. Example to of EPSP. Sige, I will not, ano na rin. Sige, just be familiar with the idea na kapag EPSP, it would want an action potential to happen. So for that, basta ang gusto niya, more positive inside. Kasi that's how depolarization happens, di ba? If the potential becomes more positive and less negative. So these examples, ang gusto nito is more positive ions, specifically sodium actually, to come in the intracellular. Pag inhibitory naman, on the other side of the spectrum. So ito naman, it brings the membrane potential further from the threshold. Or it can also maintain sa resting membrane potential. Basta as long as ayaw niya ma-excite, it's either magpapadown siya or stay ka sa resting membrane. Ayan, it decreases the likelihood of generating an action potential. Okay. So, kumbaga, practically speaking, it's not every time na you would like puro excitation. There, sometimes the signal is to inhibit, to stop. So, it will depend on the action of the neurotransmitter if it will excite or inhibit. Or yun nga, depende if what, depending on what transmitters are yung nagsisignal. Hirap. So for example, di ba, it's not every time naman na excite. When you're sleeping, you are not speaking, di ba? So kumbaga, inhibitory postsynaptic potentials ang ini-elicit kapag hindi naman ikaw magpo-perform ng action, kung hindi kailangan ng excitation. Ito, classic example also. I want you to appreciate kasi ang galing talaga nito. So like for example, 'di ba, sa flexion of your forearm, if you remember yung sa general anatomy 1 din natin, na when you flex your forearm, ito nagko-contract, ito nagre-relax. Kasi if they both contract, 'di ba, there no action will happen. If this contracts, dapat the other muscle na opposite niya will relax. So, just try to imagine na kapag nag-flex ka, dito, excitatory potentials ang dumadating kay nerve that would innervate your biceps. Okay? Nalimutan ko na yung specific nerve, but, you know, review that chart. Okay, so ganun siya. And then on the other note naman dito, since you want this to relax for the action to happen, dito naman, inhibitory potentials ang nag-reach dito sa muscle na to so that no contraction will happen. Okay? So excite, inhibit. So that's just a classic example kung bakit hindi laging excite, excite, excite. It will depend on the action, it will depend on the process, what would you want your body to do. So ganun siya. So this is just an example of inhibitory. So dito naman, you would want to maintain yung resting membrane potential or you want to make it more negative than it is. So you inhibit, you don't want the threshold to be reached. So this is just also an example. For example, ito yung chloride mo is a negative ion. So it will, kumbaga, ito ay an example of IPSP wherein nag-open yung chloride channels for the negative ions to come in the cell. Kasi you want your membrane to maintain negativity. Sige. I will not ask you to memorize the specific examples. Just understand the principle behind. So dito, like for example, ang ano dito, yun, nagsistabilize lang siya kasi leak channel siya. So kapag may pumapasok na negative, may lumalabas din na negative. So that means no change happens. So nasa resting membrane lang siya. Nagka-cancel out. Yan. So this is what I've mentioned earlier, no? So yung mga color blue, those are EPSPs. Tapos yung color red, those are IPSPs. So marami yung synapses. So lahat to axodendritic. Okay? Axodendritic yan lahat. So as you can see here, from negative 70, excite, oops, may nag-inhibit, may nag-excite, nag-inhibit, excite, hanggang sa na-reach ang threshold, action potential. Tsaka siya magpropagate along the axon. Di ba? Tapos, take note pala for this slide, axon hillock. This is where the summation happens. Okay? So, kumbaga dito kumbaga sa algebra, no, this is where you add and subtract everything that it collected from the dendrites in the soma. So, lahat ng axodendritic synapses, tsaka axosomatic synapse, EPSP man yan or IPSP, it will all be summated kay axon hillock, and then from there, it will determine kung may action potential bang nabuo because it reached the threshold or no. So, doon. Yan, doon. So, yan. The soma of the neuron acts as a sort of integrator. So, siya yung class rep. kung kamusta, tuloy ba ang field trip or hindi. Depende sa nagbayad ng field trip fee. Parang ganun. Okay. Axon hillock. That's the exact place or exact portion ng neuron where summation happens. So this is the last part. Yeah, neuromuscular junction. So this is the specific term ng synapse in between a nerve and a muscle. So ganyan siya. So, ito yung parts. You have a motor neuron and a muscle fiber. So, kapag motor neuron, it's for contraction. Kapag sensory neuron, ano lang yun, for touch or for pressure, taga-sense lang sila. Pero for this one, yung specific natin na ita-tackle is that of a motor neuron and a muscle fiber. Kasi ang next topic natin, yung unang topic natin for midterms would be muscle contraction. Wow, alam mo, exciting. Exciting talaga. Excite kayo. Yan. So, yan, in specific terms. So, yung axon terminals for neuromuscular junction, ang tawag, synaptic botons. Ganyan talaga, botons. Tapos, yung post-synaptic cell ng muscle fiber, you call it end plate. So, ganyan yan siya. So, you have your synaptic button, then you have here a synaptic cleft, and then you have here the postsynaptic cell na may mga receptors. So, as you can see here, check ang presynaptic, check ang postsynaptic, check ang synaptic cleft. So, what is this type of synapse? Electrical o chemical? Chemical. Very good. Yan. So, ito, you have here your synaptic vesicles. Tapos may mga nag-duct. Tapos ito, actually ito mga mitochondria. Ano nga ba yung mitochondria? Powerhouse of the cells. So, kailangan din siya sa axon terminals. So dito, specific din na kailangan yung alalahanin for neuromuscular junction, the neurotransmitter that is used or found sa neuromuscular junction is called acetylcholine. Automatic yun. Pag neuromuscular junction, pag nerve to muscle, automatic yun ang neurotransmitter is acetylcholine. So automatic din na for the receptors ng postsynaptic cell or ng end plate are acetylcholine receptors. Automatic din siya. Yan. So acetylcholine, EPSP, so, ganun din yung mechanism niya ng synapse kanina. Action potential reaches the synaptic button. It will open the voltage-gated calcium channels. Tapos, influx of calcium. Calcium will signal synaptic vesicles to duct and fuse sa plasma membrane of your synaptic button. And then, exocytosis ng acetylcholine. And then, may diffusion sa synaptic cleft. And then it will bind sa acetylcholine receptor. So ligand-gated yun. So it will open and then it will cause the influx of, ito pala, I have to take note pala, yung acetylcholine, okay? Yung acetylcholine, yung neurotransmitters, they are just the means for the ligand-gated channels to open. Pero what enters the channels are actually sodium ions. Okay? Sodium ions pa rin. And yung sodium ions na yun are positive ions and it will cause either EPSP or IPSP. Okay? Excitatory or inhibitory postsynaptic potentials. Basta yun na, take note, it is not the neurotransmitter na nag-e-enter kay postsynaptic cell. They are just agents to open the ligand-gated channels for sodium ions to enter the channel. Okay, this one, take note of this, acetylcholine esterase. So kapag, when you encounter ACE, okay, these are enzymes. Diba? Wow, biochem. So when we talk about acetylcholine esterase, so this is an enzyme that will break down acetylcholine. Yan. Acetylcholine is removed from the synaptic cleft by acetylcholinesterase, which is present in high concentration at the neuromuscular junction. Action potentials are generated on either side of the end plate and are conducted away from the end plate in both directions along the muscle fiber. The muscle action potential initiates muscle contraction. So, basta yun. Yun yung pinaka-action of acetylcholinesterase. It will clear up the synaptic cleft from acetylcholine. So any diffusing acetylcholine, kumbaga yung hindi naman nag-bind sa receptors na nagfo-floating lang sa synaptic cleft, it will be captured and kumbaga may enzymatic activity by acetylcholine esterase to remove those excess acetylcholine. And actually, nire-recycle back yun for the neurotransmitter uptake. So this one is just a teaser. Teaser. For midterms na. Okay? Midterms na lang. So, ganyan siya. May alienated axon of motor neuron. Action potential reaches synaptic buttons or axon terminal. Tapos, ito yung end plate na nag-receive, nag-post-synaptic cell for the synapse. And then, there are happenings that happens inside the muscle fiber based doon sa action potential na nakarating that reaches the muscle fiber. And then from there, we will discuss next meeting how muscle contraction happens when the innervation reaches a muscle. Next meeting na lang. So ngayon, focused lang tayo sa synapse and synaptic transmission. Ito, maganda to. Yan, acetylcholinesterase. Kita niyo yan. So that is present sa postsynaptic cell. And it, yun nga, enzymatic activity na dinidivide niya si acetylcholine to acetic acid and choline for recycling. Yan. So, kapag may ligand-gated channels, pasok si sodium, labas si potassium. So, whatever action potential happens here, EPSP ba or IPSP? That's it. So, yon. Ayan. So, that's our lesson for prelims. Okay, membrane transport, membrane potential, action potential, and synapses and synaptic transmission. So if there would ganito ha, since asynchronous ang general physiology natin and you are the same set of students sa general anatomy 2, okay, I will also give you the privilege na rin to clarify things, to ask questions about general physiology during our synchronous meeting sa Gen Ana. Pero sige, I will make a delineation. Bigyan natin siya ng time frame na for example, I will post it sa MS Teams natin na like for example muna to, I will think about it pa ha. So for example, 9 to 9:30 we'll talk about general anatomy 2. So may graded recitation tayo, ganyan-ganyan, clarifications and Q&A kung may questions kayo, whatever. And then next part, para since I'm already engaged with you, ganyan. So next part, if you also have any concerns with general physiology, we can also tackle it during that time frame. Okay lang. So basta Thursday is our synchronous day. Okay? For general anatomy 2 lecture, general anatomy 2 laboratory, and general physiology lecture. So the I will also mention na rin at that time the orientation as to how your exam will be, and that would be next week na pala. Okay? So I hope you learned today with our lesson, review the lesson, and if you think you're prepared enough to take the quiz, then it's also available here sa ating LMS. So thank you and see you tomorrow if you watch this video on a Wednesday. I'll see you on a Thursday. Bye. See ya.